Today I will be talking about some work that we are doing in my research lab here at University of Prince Edward Island. The title of the talk is Self-Assembled Peptide Nanoparticles. My name is Marya Ahmed. I'm a professor in Department of Chemistry and Sustainable Design Engineering. So why peptides and what interested me in this work? Recently, you may have noticed there have been a lot of recalls on food, and this is just one example, salmonella infection linked to raw chicken, and there have been many other incidences over the last years where a lot of food recalls have been issued, not only on poultry, but also on other meat and other products. But poultry has been a major component of those recalls. So first, I'm just going to talk about what's the background of these recalls and what we are doing to fix this problem. Of course, this causes a tremendous loss of economically to the country and also to the food producers and the general public who, if by mistake, consume this food, this becomes a major health issue. So all this kind of started with antibiotic growth promoters, and it was a time of 1950s when there was a growing demand of meat, including chicken and beef and other types of meat, to meet the rising demands of general public, the increase in population and all that. So the easiest way found was if we can use antibiotics at sublethal levels in the food of animal, this was found to improve the growth of the animal. By adding the antibiotic, this suppressed the infections of the bacteria or the microbes that were present in the gut of the animal, and thus increase the meat production and we could actually meet the demand of the public consumption of the meat. Slowly, this started out really well, but slowly it was in 1990s when this became quite obvious that the antibiotics that are being used in animal husbandry or animal growth are becoming less and less effective to human. So antibiotic resistance started becoming a problem in 1990s. Just because we got used to eating the antibiotics that became part of the food that in the form of a meat. It was around 2006 when European Union issued a ban on antibiotic growth promoters, and it was not until 2017 when US FDA issued a ban on antibiotic growth promoters, realizing that this is increasing the resistance in human. So this was the time to start looking at other alternatives to maintain the high production of meat while improving the animal health and also the overall safety of the food. So some of the alternatives that were considered were bacteriophages, engineered peptides. This is what we will be talking about today. Antibodies, probiotics, prebiotics, and other small molecules. So the topic of today's talk will be engineered peptides to kind of overcome this drug resistance issue and to produce meat that can be safely consumed by humans. So what are antimicrobial peptides or proteins? So antimicrobial peptides and proteins are generally present in all living things, including animals and human, where they are produced by the own immune system of that animal or the human. And the role of these peptides are multiple, but one of them is that they can kill the bacteria, they can kill the microbe, and overall, they can just dissolve the biological membrane of a microbe, hence killing them. But this is just one of the many mechanisms. These antimicrobial peptides and proteins have many other roles. They help with the neutralization of toxins. They help with immune modulation and activation. Of course, they directly kill the bacteria as well, which I've just talked here. They also participate in wound healing and many other mechanisms. So if they're already present in human or animals, this would be a lot easier to use those naturally present proteins in given animal and mimic those to improve the food safety. And that's what we did in my lab here at University of Prince Edward Island. Working with a poultry group at Atlantic Veterinary College, we started looking at chickens because poultry is one of the most consumed meat overall in the world and also in North America. And we found that in poultry, there is a protein which is called angiogenin 4, Ang4, which is highly expressed in the last part of the intestine we call bursa. Chicken angiogenin 4 is responsible to maintain the health of the animal gut. And there is quite a bit of data which is also developed here at Atlantic Veterinary College in PEI, which shows that if this protein is in high levels in a chicken, chicken overall has a good health and less infection. So we started to take this protein as our template to engineer the peptides. And I'll go there in a minute why we can't just use the protein. So chicken angiogenin 4, it's a big protein. It's about 16,000 Dalton. This was first discovered in 2012 in Professor Rodriguez Lecomte lab in, here at Atlantic Veterinary College of University of Prince Edward Island. Its overexpression really helps with minimizing the pathogen infections in chickens. But the issue is it's a big protein. And protein, people who work with the proteins, they know that expression of protein is highly tedious. It's not an easy task. The yields are very low, and also it quite varies from animal to animal. So we thought that if we take this protein as a template and figure out the key components of that protein, we can make some peptides. And with the growing field of peptide chemistry, it should be a lot simpler method to develop small peptides, which we call mini chicken angiogenin 4s, that may have better or similar activity to the whole protein. And then we can inject our peptides in the poultry feed and help the chicken health. We took this protein as a template and we made fragments, which we call mini chicken angiogenin 4. The idea of developing these fragments was that the fragments will be much smaller in size, can be easily made by solid phase peptide synthesis, something we do in my lab here at University of Prince Edward Island. And since they're much simpler to make, we may expect that they have similar or even higher potency than the whole protein. We developed those peptides by a combination of catalytic triade sequence and cell binding sequence of the whole protein, and by truncation and residue substitution and introduction of thiols to improve the cellular uptake and antimicrobial efficacies. You see here, there are 5 sequences of these peptides, MCA4-1 to all the way MCA4-5. So once we made them, we characterized them and we looked at their antimicrobial properties. So here you are seeing ampicillin we used as a control, chicken angiogenin 4 itself, the protein, and the 5 peptide sequences that we derived from this chicken angiogenin 4. So you see here, chicken angiogenin 4 is a big protein, about 16,000 Dalton in size. The peptides are pretty small, ranging from 700 Dalton to 1,200 Dalton. So they can be prepared easily and purified. And then we looked at their antimicrobial efficacies and dominant pathogens present in poultry, like E. coli, Salmonella, and Listeria. So here you see ampicillin, that is a traditional antibiotic, does have reasonable activity against these pathogens. Chicken angiogenin 4 protein itself is mainly active in Salmonella and Listeria. Most of the peptides we studied did not work well, but there was one sequence that we said MCA4-5 was highly active and was also active in bacteria that were resistant to original chicken angiogenin 4. So you see the activity is similar for Salmonella for chicken angiogenin 4 and MCA4-5, and Listeria, it's slightly higher, but the peptide is also active in Staph aureus and E. coli. So this gave us preliminary success data that we have a sequence of peptide derived from chicken angiogenin 4 that works really well against multiple bacteria. After that, we wanted to see that if we take some bacteria and infect chicken cells with that, in this case, we took intestinal cells of human, would the peptide be able to control the infection? So here you are seeing cells attached, and what you're seeing, those dots, green dots, they are the bacteria. You see there is a lot of green dots where there is untreated cells in the presence of bacteria, no peptide, no antibiotic. A peptide, chicken angiogenin 4-4, did not work well, and you see a lot of bacteria present around the cells. Whereas MCA4-5 that worked really well shows very few dots, showing that most of the bacterial infected cells were killed. So this indicates that the peptides we made has ability not only to kill the bacteria, but also to cure the bacterial infected cells. Next, we wanted to study about the secondary structure of these peptides. So peptides, mostly if they are of certain length, more than 10 amino acids, they have tendency to self-assemble into different forms. So we started doing a transmission electron microscopy, and what we saw was, to our surprise, these peptides are making these small spheres of about 10 nanometer. So we set on to look at the structure, and we found that the peptides, which were prepared as the random chains, once put in solution, fold themselves into beta turns and beta sheets. And these beta turns and beta sheets then self-assemble into small spheres that we see in this TEM image as the dots. And we named them as a disulfide linked nanospheres, or DSNs. One of the critical components of any peptide nanoparticles are that they should be stable. Because if we want to use them in chicken gut or for human health applications, if the peptides are not stable, they will aggregate and degrade over time. So we started looking at the stability of our disulfide linked nanosphere. Here you're seeing dynamic light scattering analysis. Here, pay attention to this line first. These are our disulfide linked nanospheres that are showing the size of 250 nanometer. But from our previous slide, we know that they're only 10 nanometer. So this indicates that our peptides are aggregating, showing much larger size than they actually should be. Also, when we put these nanoparticles in physiological conditions, in the presence of salt and some serum, you clearly see in the photograph that they are becoming turbid, and the size increases up to 5,000 nanometer, clearly indicating that disulfide linked nanospheres we have prepared are not stable under physiological conditions. So to overcome this problem, we looked towards sugars. The main reason we wanted to stick with sugars that were naturally present so that we can easily aid our nanospheres for any future work towards health applications. So we use fucoidone as a one sugar that is a type of a seaweed. And the second is beta-cyclodextrin, a sugar commonly found in many fruits. So when we coated our disulfide linked nanoparticles with fucoidone, this did not help us. You can see here the fucoidone nanoparticles show rapid increase in size over time, showing poor stability. And same is seen right here, rapid increase in size, showing instability. Whereas when we capped our disulfide linked nanosphere with beta-cyclodextrin, we see very small, about 10 nanometer sized nanoparticles that were maintained and were similar to what we saw here in TEM analysis. So by capping our disulfide linked nanoparticles that were aggregating in solution with cyclodextrin, beta-cyclodextrin, we were able to produce well-developed nano-sized peptide-based nanoparticles. Once we did that, we wanted to know more about the structure of those nanospheres. So we said earlier that our peptides fold into beta turns and beta sheets and beta turns. And we can see this by this ATR FTIR profile, where the peptide alone has this distinctive peak, a very strong peak at 1630 that is indicative of beta sheet. Whereas this broad peak in the red region is parallel beta turns and beta sheets. Capping of our peptides with cyclodextrin kind of reduced the secondary structure, and you see the reduction in this secondary structure formation in the presence of beta-cyclodextrin. So in the absence of sugars, the peptide folds into beta sheets and beta turns involving disulfide bond, but stabilization of these nanospheres with sugars causes a loss of antiparallel beta sheet structures compared to the peptide alone. So we wanted to know then that if the secondary structure of peptide is changing, what is going on with the activity of these new nanoparticles that we have stabilized with sugars. So we looked at antimicrobial properties. So again, ampicillin was our control. That's a traditional drug, active against most of the bacteria except B. subtilis. Disulfide linked nanoparticles that were in the absence of sugar showed good activity, that what we have seen before. But actually the capping of these disulfide linked nanoparticles with beta-cyclodextrin improved the activity of our nanoparticles. Although there was a loss of secondary structure, overall activity improved quite a bit. So look at E. coli, 15 micromolar to 3 micromolar. Similarly, Listeria, several-fold improvement in activity just by capping our nanoparticles with beta-cyclodextrin. So these results were quite impressive, and we wanted to look further into the mechanism of bacterial death in the presence of our peptides. So here in this photograph, you're seeing bacteria alone. Live bacteria, we stained them as green. And you see in PBS or a simple physiological solution, bacteria are well dispersed. As soon as we add our disulfide-linked nanoparticles, all these bacteria aggregated and settled down. And if we take those aggregates and we try to find live bacteria, we see very little live bacteria and most of the dead bacteria. The red color here is indicated for dead bacteria, which are not present in the control solution. Similarly, when we added sugar-capped or beta-cyclodextrin-capped disulfide-linked nanoparticles, we saw the same trend. All the bacteria crashed out of the buffer and there was no live bacteria found. All the bacteria that were trapped in this cluster were dead bacteria. So overall mechanism to this was that the disulfide-linked nanoparticles and their sugar analogs can trap the bacteria and kill them by membrane permeabilization. Another important property of nanoparticles is that they should be toxic to the pathogen, but not to the normal cells. So we did a red blood cell lysis assay, and we found that Triton X100 was used as a control that can give 100% blood cell lysis. Compared to Triton X100, all our nanoparticles only showed 4 to 6% red blood cell lysis. And also our nanoparticles were non-toxic towards intestinal cells and chicken fibroblasts, indicating the safety of these new peptide nanoparticles towards combating bacterial infection in chickens. So overall, we have looked today a type of peptide nanoparticles that are derived from chicken angiogenin 4. We call those peptides mini chicken angiogenin 4 that self-assemble into nanoparticles, and these nanoparticles and their bacterial aggregation is considered key for their peptide activity. Also, the nanoparticles produced are non-toxic towards chicken cells and blood cells and have the potential to cure bacterial infection in mammalian cells. That's all I wanted to share today, and I'm happy to answer any questions. Thank you.
Marya, thank you so much for sharing that. And I believe the numbers of obviously chicken that are bred every year and also that are slaughtered is very, very high. We're talking not millions, but billions.
Yes.
And bacterial resistance is what you mentioned right at the beginning is an issue. How is it with resistance for antimicrobial peptides? Do we have more potential there that they will remain effective for longer? Or can we also, do we have more opportunity also to modulate them to adjust for antibacterial resistance?
That's a very interesting question and one of the major field of work right now that many people are working on. So if we understand the mechanism of antibiotics, the antibiotics usually target molecules inside the bacteria. For example, they inhibit protein synthesis or DNA synthesis or any downstream, whereas peptides, they mainly act on the membranes, on the bacterial membranes. So for bacteria, it's a lot less costly to kind of prevent one machine from working. Mm-hmm. But having the whole membrane modified, that's hard for bacteria. So for the antimicrobial peptides, their potential to develop microbial resistance is much less than antibiotics. Yes, this happens. Some bacteria tend to over or underexpress some receptors that may have negative charge and will allow the attachment of peptide more easily compared to the others. But this is much, the rate of resistance is much lower than antibiotic. It is expected to be much lower than antibiotic, simply because it's much harder for bacteria to pretty much change their whole surface as compared to one protein or one mechanism. That being said, it also depends on the type of peptides. Some peptides have the ability to develop resistance, some don't. And that depends on the overall structure, their net charge, and other properties.
Marya, thank you so much again for your time, and
thank you.
Best of luck with your further work. Thank you.
Thank you. All right. Bye.